Alumni Affairs, who has given me this opportunity to interact with my alma mater. I'm very pleased to do this. And I would especially like to thank Bhargavi ma'am, who has coordinated all of this, and Angeline ma'am, who actually put me in touch with Bhargavi ma'am for giving this talk. So today's talk, I have divided it into two parts. The initial part is very generic. I just wanted to give an overview of what are the career choices you have as a biomedical engineer, and especially more focused on the research side though. And then in the second half, I will give you whatever I explained in the first half, I will give you an example of that through my research and how it went. So for the first half, you all would have a lot of questions probably. It might be nice to take it at the end. But for the second half, where I'm talking about my research, I would want you to interrupt me and ask me the questions. Otherwise, it might be difficult for you to follow. Okay, so I am hoping I don't see anybody or I can't hear anybody as of now. So I hope if there is anything, just interrupt me. Yeah. Okay, so the topic is to analyze or to synthesize. You will understand the meaning of this as I go through the slides. Okay. Yes. So biomedical research in general is focused on a wide variety of areas. So it includes very basic research where you want to understand some physical, chemical, or any other functional phenomena or mechanism of all those biological processes in humans and animals. Then there is another phase to it where you are actually, this is in a normal scenario that you do it. Then there is a phase to it where you actually look at cases where it's not normal, it's abnormal. And then only after understanding that can you actually figure out how to prevent it or treat it. Then there is an entirely different side to it, which is designing or inventing technology or devices that would be useful in improving the healthcare. So all of this can be broadly put into biomedical research. Now, so as I said, it's a confluence. It's like, so it's a confluence of three major things. This is something I believe. So one is basic research, as I said, where you try to understand in case of health and in case of disease, how some biological processes are happening. And then in case of disease, you need little more specialization from the clinical side, because you need doctors to give you input and stuff. So there is clinical research has developed on its own in a different way as a different regime. And then there is applied research, as I said, in the third category, where you are developing technology, where most of the engineers are focused on. All of these come under the biomedical. So sometimes what happens is, it's not just, so for example, if it's a basic research, pure physicist can still do that basic research if he understands a little of the other parts. A doctor who is not aware of engineering or technology can still actually do biomedical clinical research. An engineer without knowing the rest of it can do an applied research. Engineer can build circuits that can be used in healthcare by just understanding stuff. But then it becomes very effective and productive when you have an interaction, an ecosystem where all of these specialists interact. And biomedical engineers have an upper hand because they themselves are that ecosystem. You know the physics, you know the chemistry, you know the biology. We have a flavor of all that in your biochemistry or any other courses. You also know the electronics, the mathematics, which is required to actually do all the simulations or understand the system or develop products. You also know how it varies. You also have situations where you learn about illnesses and stuff. So as that is where we all biomedical engineers, I'm hoping, because we are all being trained in biomedical engineering department, that's where we have an upper hand. And interdisciplinary research, which actually makes a collaboration between all of these, is very important for the future, because there are a lot of people working on all individually in each of them. but then only if we have a collaboration can we be very productive in handling health issues. So I broadly divide the area of biomedical research into the science and the engineering. It's a very broad division. It's not clear cut. There might be interactions and stuff. But then what I mean by science is where you analyze things. That's my so if you go back to the topic I had, to analyze or synthesize. So the science is the analyze and the engineering is the synthesize. So when you are doing a scientific research, what you do is you break down the things into smaller bits and the whole system, and then try to understand each bit and then get the big picture of that, of the entire process that is happening. And the methodology that is followed is something like this. And this methodology, this slide is I will make you understand this with an example through my research. Okay, so initially the idea is to ask some question. You want to know the answer to some question. So you go do all the background research, what have other scientists found, what has been observed, reported, and everything. And then you construct some hypothesis. Oh, if this was, so if with all the information you've collected, you say that for this question, probably this could be the answer. That's your hypothesis. Now you need to test your hypothesis. So you need to design your experiment. This actually is a very critical stage, and it's I think one of the most difficult after finding the question. Finding the question is the most difficult part. Finding a unique question or the right question, and then the difficult part is to create the right experiment to actually answer that question. Then once you have the experiment working, if it's working, you can analyze the data, observe what is happening, and draw conclusions. But if it doesn't work, you have to go back, and this loop can actually take you a lot of time. Then once you have the conclusions, sometimes what happens is, yeah, your hypothesis was right, but sometimes it is not right. In both cases, you communicate the results to the world so that someone else can go beyond it or use that like we did use the background research. Now, this is a special loop where you have the results and conclusions. Now, this becomes actually the background research for defining a new question or a new project. In many of the PhD labs, this plays a big role, and in my case, I believe it did the same way. I will address it when I come to that particular place. On the other hand, it's the engineering is more of synthesizing, where you put together. I will explain the procedure later. So, but then the idea is to put together small building blocks and develop something. You create something that is very useful for healthcare applications. So there, you same way, instead of question, you are defining a problem that is faced, and then you do all the background research, and then you see what are the requirements to address this problem. Then you brainstorm, evaluate, combine some already existing solutions, or come up with an out-of-box new solution, and then you try to develop and prototype that solution, test it. If it works, well, good, you communicate it and use it, market it. If it doesn't work, then you go back and reiterate on this process. Okay, so that are the two general classification I believe that is there. And it's very important that you make the right career choice, because you have a broad spectrum of choices. People are always confused. So I think there are the choices can be broadly divided into an academic teaching kind of thing, which many prefer. In that case, after your MTech, BTech, or MTech, you actually get into teaching. You like teaching. Teaching is also so there you don't have much focus on research. You do research as part of, say, for example, training your BTech student, taking up your BTech student projects, or students projects, or MTech students projects. But then you do not get into deep research. Then there is academic research, where you do very little teaching, but you actually train PhD scholars and have your independent pipeline of research where you want to explore a particular question. Then the next option is industry. In industry, mostly in India especially, it's more concentrated on product development, the engineering side of it, and it's more on servicing, maintenance, and that for the biomedical engineers, that is the scenario. Although now big firms like TCS, GE, Philips, all of them are involved in a lot of imaging based, because the MRI imaging devices, MRI, CT, and everything are being built in India. They do have a research oriented wing which is focused on image processing and application of artificial intelligence to all these for automation of the devices. But then in general, rest of the industrial research is more abroad, where, for example, in case of Medtronics and all their devices, the research level happens abroad, and only the assembly and the product development kind of thing happens in India. So those are two options in the industry. Then there is a completely different domain where many of us end up going after the BTech, like, because we are trained in programming, everything, most of the basic things required for an engineer, any IT field or a software programmer or data scientist, any of those fields can be chosen by us. It need not be biomedical, because the training actually helps us have all the skills that are required by those industries. And then there is, it's not nonsense, it's a non-science domain, which is something pseudo. So there is, so as biomedical researcher, it's something what I work now. For example, in my company at Alien Technology Transfer, what we offer is project consultancy. So there are startups which have some new idea and they would like to get funding. So in order to get funding, they need to actually do detailed analysis of whether the technique is feasible, whether how should be their business strategy, how should they actually how do you collaborate with people on all those things. So I don't know if you guys know, there is management consultancy done by a lot of companies like Deloitte, McKinsey, and all. So there is a science wing to it where you need a little technological knowledge also. So that is also a very good domain where you can be involved in the non-science aspect. You get to develop more of your entrepreneurial skills. There is another whole domain of the healthcare industry. It's just not engineers and product developers. You need a lot of good managers and hire people who have an understanding of the healthcare domain and the technology a bit, but then are good at managing the business. So you could go in that direction also. So depending on what each of these demands or what each of this will provide you, you need to make the right career choice. It's you need to do a good analysis to make the right career choice. Otherwise, it may not be satisfying as a human being. So here, I am mostly in this domain, academic research. I tried this a bit for a year, and I am now here. But then I decided that this is academic research is something that is of interest to me. So I will give you through an example the science scenario which we discussed before. Through the research I did for my PhD training, and it's a program at Indian Institute of Science, a Center for Biosystem Science and Engineering. It's an interdisciplinary program where I had a guide from engineering, a thesis advisor from engineering, as well as someone from neuroscience, and we together actually tried to solve a particular question. So the question that we identified was saccadic eye movements. I will tell you what saccadic eye movements is. The question was to understand how these eye movements are made by the brain. And then the approach we used is an engineering approach where we used control systems theory. I'm hoping some of you at least know about that, to understand how these movements are made. So, so the technical part of the talk starts now. So if you have doubts at any point, just interrupt me and ask me. So what are these saccadic eye movements? When you are making, So when you are making very fast eye movements, so when you're looking at the face of a person, you keep making a lot of eye movements, which you are actually unaware of. These fast eye movements are called saccadic eye movements. Okay, and So if you plot with respect to time, how one movement happens, it's something like this. It's at rest, the eye is at rest, 0, there is no displacement. And then from some center, it moves very fast, and then it lands somewhere. So say you are looking from one point on the screen to another one, like say you are looking from here to here. Basically, the eye fastly moves and then stays there. And if you look at the corresponding velocity of this movement, it increases very fast. Because you have to stay there, it starts decreasing and then settles. So this is for when you are making something around 10 degree. This is when you're making, I say degree because it's an eyeball. You have an eyeball, and you are talking in angular dimensions. And then 30 and the corresponding velocities. Now, how does the brain generate these movements? So I went up, as I said, I have a question. I want to know how these eye movements are made. So I went and did my background research, as I said. So what people had figured out is that a lot of areas on the frontal areas in the brain, a lot of areas in the brain actually contribute to telling us, oh, you have something to see. For example, you want to pick up a cup from the table. Oh, so there is a cup there, and that is at some distance from your hand. So your hand is here. So all this information is processed, and then finally there are some areas in the lower brain, the inner parts, not the surface, and those areas which we call the brainstem area. Those area actually you give the higher areas. These are called higher areas. The higher area gives the information. It processes everything, hand, cup, and everything, and says that the eye needs to make a movement of say 10 degree. So that information is given here. And then this area actually creates or generates signals which needs to be given to the eye muscles. And when those signals reach the eye muscles, a movement is made. That is how. So people have already understood that this is how eye movements are made. This is understood by a lot of studies where they record from each of these brain areas. They do modeling, they look at, do experiments and learn all this. Now I said my approach was to look from a control system perspective, right? So for a control system, I convert this brain areas and everything into something simple, just blocks, like block diagram. That's the first thing we learn in control system subject. I think you make block diagrams and you learn on how to reduce that block diagram into one single equation of a transfer function or something. So it's similar approach. You convert all this what you see here into a block diagram. So these areas provide you the plan, and this area, so the plan is, say you want to move like this, and then this area actually provides you a burst. I'll tell you why a burst, because the eye has a lot of mass, right? So initially to start displacing the eye, you need a high signal. That's why the fast rise of the signal. And then once you're almost there, you can just settle down and it comes down. So this is kind of similar to the velocity. So that signal is encoded by this area, and that is given to a pulse step generator. Why the step? This is the pulse part. Now you need a step because if you don't give a step, the eye, because it's elastic, it's just going to come back to its initial position. So to avoid that, you constantly keep giving a signal so that the eye remains at the cup which you are going to see. And then the oculomotor plant is the muscle. So this is the signal that goes to the muscles, that activates the muscles, and the eye moves. So if this muscle gets a signal, the eye would move this side. The other muscle gets a signal, eye would move the other side. If this muscle gets a signal, it moves up, and this muscle gets a signal, it moves down. Now it can also move in oblique direction, which is an action of all muscles combined. So this is, so if you keep doing this, sometimes what happens is it's not very good, right? So it may not, you gave the signal, but the eye may not have exactly moved that way. So you need to know whether the eye is moving correctly. So as per in usual control systems like fan or anything, what you do is you know how it moves, and then you use that to correct the signal. But in case of saccadic eye movements, this particular type of movement, what happens is the movements are very fast. So for example, I am talking about a movement that is happening 50 milliseconds. But for the brain areas to process all the information, it takes 70 milliseconds. So you do not have the time to at every time point to check whether the light did move or not. So what is hypothesized is that what the literature told is that this signal, you take a copy of that itself and use that to make sure the difference is not. So this is exactly what you want. So the plan here is perfect. And then you have a signal coming from here, which actually checks what this information is, and see that it aligns perfectly with the plan. So this block, what it does is predicts everything that happens here. So it predicts what will be the outcome of this, because it knows what is happening here. It calculates that and gives this outcome by itself and checks that everything is going right. So instead of a normal feedback which we have learned in control system, it's an internal feedback idea. So this is the general Saccade Control. Do I have any questions? Okay, I'll take that as no. So the Saccade plan here. So when again I'm digging into the literature. Okay, the Saccade plan here, interestingly, some of them suggested that the plan here is the desired input signal is a so everything is in firing rate because it's neurons encoding them. So forget that. For now, we will just think that it's a step signal. So for example, if you want to go 10 degrees, so then you just say, oh, you go 10. So every time what happens is it's saying 10. And then when this signal is generated and this calculates everything, oh, it has only moved 1. So there is 9 left. So again, it gives a signal, oh, it has moved only 2. So then there is 8 left. So this keeps happening till the 10 is reached. So when the 10 is reached, you reach the endpoint. That is one kind of model that, or one kind of theory of how the Saccade control is done, that was suggested in the literature. This we will call displacement control, because what only thing you suggest is the displacement, 10-degree displacement. But then there was another set of studies which suggested that it's just not displacement. At every time point, you tell them how to go. You go first 1 degree, then you go 2 degrees, then you go 3 degrees, and then you go slower. So you could encode the entire trajectory. It's just not the final 10 amplitude that is done, but you tell it every time point, oh, this has to happen, this has to happen. In both these cases, the feedback which comes would change, and this block, the burst. So in the previous thing, the burst, the function that is done by the burst generator also would change. So now there is a confusion. Oh, which one is it? Is it displacement or velocity? Oh, and so what I did, so as an engineer would have done, I simulated both of this. So how would you simulate that? The next 3 slides, don't get afraid. If you don't understand anything, it's okay. So what basically is done is you actually write equations for each of those blocks, what they represent as a function, and then you give those equations. There are all steps has equations. You represent them using mathematical equations. You simulate that. You code those equations into MATLAB, and then you generate what it will produce at the end of the model. So when I looked at displacement model, the model predicted these red dots, and I had some data of eye movements from some 20 people. So I actually fitted the model to that, and it predicts exactly what the data is for all of them. And if you look at the velocity model, oh, it also predicts everything. Now what? Both of the model gives same prediction. So that's when, like I said, in the approach of scientific thing, there is a feedback loop where previous results help you form new hypotheses. So that's where that vision, where in my lab with my professor, there were seniors who work, where they had found that what I showed you here is so when you know that when a person makes a movement, so I'll go here. When a robot makes a movement from the start to 1, 2, 3, or 4, they exactly make the same movement every time you tell it to repeat. But now, if it was a human, every time you draw the same straight line, it might go here and there a bit, right? a batsman cannot exactly hit the same trajectory every time. So there is a variability which is inherent, which we cannot control, right? So people, and in my lab, it was observed that this variability is an important thing. So why do I say the variability is an important thing? I will tell you in the next slide. But then why does this variability arise at all? That is because the muscle, the brain, everything we talked about is not ideal. In a robot, it is very, it's mechanical. It doesn't change. Every element is same. But then when you go to people, each person is different. Within a person, every time a muscle contracts or a brain fires, it's different, right? So that difference or that small change is the noise. And but if you repeatedly look at this information and then you average it, average of all the times you repeated it, that is what I showed you in the previous slide. It's like the average data. So in literature till now, people had looked at only the average data. They did not care about how it varied. So the idea that came up from the lab is that how it varied also is important. So that's how so now, because I was stuck, there were two models in the literature. Both of them talk the same thing about the average. So now what? How do I distinguish which is the model? So that's when I used this idea of variability. And why I think it's important is that, say, for example, you are throwing a dart to the center of the screen. So initially you are very new to it. Then you will do it all over the place, right? All these dots are where you threw the dart. And then as you become an expert, what happens? Everything falls at the center circle. So from a novice, a new, like beginner to an expert, this changes. The variability actually reduces. In the same way, when you go from a healthy to a patient, when you are a healthy person, the variability is there because it's natural to the system, but it is less. But in a patient, it's more. So these things suggest that the variability has something to do with the noise in the system, and when you generate movements, there should be an active control of that noise. So there is a possible active control of the noise. That is what actually makes sure that the noise in normal people is less, the effect of noise, but in the noise in the patients are higher, and the effect of noise. So we thought that maybe this could be used as an idea to study in Saccade control. We are going to actually then look at the intertrial variability in the Saccade trajectories, and then understand how the control is happening. Any questions, Amira? Okay. Hello.
Hello. Somebody.
Any questions?
No, I think someone said hello.
That's why I was waiting.
Okay.
Okay. We'll take the questions later then. Yeah. So how did we do that? So as we discussed earlier, when you have I have a hypothesis now. My hypothesis is that the variability in the movement will tell us something about the control or how the movement is generated. So now what you do, you go and design your experiment. In my case, it was very simple and straightforward. There are situations where it can get complicated. So what I did is, on a screen, I show a white box. The person sits in front of the screen and looks at the white box. Suddenly a green box comes up, which is called the target. So the person who is sitting and looking at the white box has to just make an eye movement to this green box. Their head, everything is constrained, so they can't move their head. Only the eye moves from here to here. So that they made us a card. And using a camera, I actually record this. And I recorded this to a lot of positions on the screen. So every time only one thing comes. That's all they see. But then this green can come anywhere in these positions. Now I did this and then I recorded the eye movements, and I have the data. So it's not one single movement. They keep making these movements to different targets multiple times. So for each of these targets, I have around 50 trials, 50 times, 50 repeated movements. So this is, so for one person with respect to time, this is how the eye move, the displacement of the eye. I move, so the target in this case was around 12 degree. I move, reach 12 degree. I move, reach 12 degree. So they kept repeating it, and then this is the repeated movement. In all the existing models, what did they do? They just looked at the average somewhere here, somewhere in the middle. But then what I did is quantify how the variability changes. So here it's very less. The spread is very less, right? I quantified the spread with respect to time. That's what I did. And so such a quantification of the spread here is shown for 20 different subjects, 20 different people who did this experiment. Okay, so here it's the time, which what shows is 100 is the end of the movement and 0 is the start of the movement, and it's all normalized. What I'm showing you here is like the x-axis is normalized, and y-axis you see that there was a spread. So this spread is what you can make here. The spread is around 0.3 here. For another person, it might be lesser. So this person it's a little more. It's 0.7. And then what's interesting is that the way it's changed also varies. For some people, it's just increasing. There are people where it's just saturating towards the end, and there were people there where it was a bit decreasing, right? And decreasing profiles. There were a variety of profiles. But now the idea is, what we wanted to do is that two models, right? Which model is it? Which way is it controlled? Is it displacement control or velocity control? So in order to do that, those models, because they look at only average, you only need an ideal model. But here we are looking at variability, which is arising due to noise. So in this case, you need a stochastic model where the noise is also added to all these blocks. So I added a version, I modified and brought in a version of a noisy model where noise affects at each stage. Now what I'm going to do is I'm going to simulate this model for both the displacement case and the velocity case. So this is the simulation, and what it predicted is that for the displacement case, the model is more careful about how much spread is there in the displacement. It wants to keep this less. So the variance increases, but towards the end it decreases. But in a velocity case, it just keeps increasing. So in case of a pure displacement model, the maximum variance was somewhere before the end of the movement. But in case of velocity model, maximum variance was at the end of the movement. So this is a profile where it increases and then decreases. But for velocity, it's a profile, it just keeps increasing. But remember, in our data, what did we see? We saw increasing, we saw decreasing, all types of profiles, right? Then we were like, oh, then what? Which is the model? Which one is true? Because we have both of them. So a direct, straightforward assumption then is to combine both. So we combine the displacement and the velocity model, and then we try to simulate the variance. So what we saw is that depending on how much of displacement is involved or how much of velocity is involved, we could predict all kinds of profiles. For example, we could have increasing and decreasing. We could have saturating. We could have just increasing profiles. So here is my prediction from the models. And what I did is went back to my data, which showed, so the squares are the data points. And then I fit my new dual model into the variance, and it could predict all of them, the increasing ones, the saturating ones, and the decreasing ones. So the conclusion, and obviously we need to make sure that these fits are better than the individual models. So we compare the dual model with the displacement and velocity model. When you look at the average, all of the model predicts the same way. There is no difference. But when you look at the variance, displacement model has a lot of error. Velocity model also has a lot of error. But the dual model has very low errors in percentage. So each of these dot is each subject or each person who did that experiment. So the conclusion we drew from this is that Saccade control in the presence of noise, which people haven't looked at. That's the novelty here. In the presence of noise, it is best explained by dual control of both displacement and velocity. So whenever needed, you can have more of displacement, less of velocity, more of velocity and less of displacement and things like that. And something that actually helped us reach here, this conclusion, is because people never look at variability. They only looked at the average of the performance. So if you look at the variability, that seems to be an important tool to understand principles of how movements are made, how the brain controls the movements. So this approach is very useful. So I will give you why this study is impactful, because say in case of patients, by looking at variability, if you can predict that, there is a chance that they will develop the disease or something like that. That kind of study would be very impactful. And then, as I said, from novice to expert, it changes. So if you can tune the variability of a player such that there is very less noise and that performance increases, for a player, it will be a very important for his career. So in sports medicine and sports related things, and in rehabilitation in diseases in patients and Parkinson's, these kind of understanding of the system might be very important. So that's the technical part. Any questions?
Participants, you can unmute and ask the question, or you can leave the question in the chat window. Actually, Varsha, this is like very interesting for me because I took this topic in biocontrol systems once.
Yeah. So, okay, by the time they think of their questions, I want to actually add something. So I
Sure.
want to tell everyone how, so something to take what I have felt through my PhD training. So basically, the PhD degree is a training for the bigger research you are going to do tomorrow. So no, it's not necessary that you are a big good researcher or you have a lot of publications and everything, but it's necessary that you have developed all the skills that are needed for being a good researcher. And if you are comfortable with these skills, only then you can actually, it's a good idea to choose research as a career. So some three important things which I feel is really necessary if you want to pursue a research career is self-motivation and commitment. Nobody is going to tell you you do this. In an industry, the management, or there are people who would decide what to do, and you do this, and then they tell you and you just do it. But in research career, you have to figure out what you want to do, how you do it, and everything. It's a lot of self-motivation is required. And then you have to always have interest and curiosity to go read what other people are doing, think beyond and critically beyond about what they have done in those things. And you have to always be up to date with your field of research. And then you need very steadfast perseverance, because every day you might fail. You might fail for months, but then again next day you should have the motivation and the determination to come back and again try on that. You should have that. You should happily come back and try on that. That is very important. Only if you think that you have these, do choose research as a career. Otherwise, it may not be your. It's okay. Everybody need not be a researcher. So you will have your own things which you make you happy. So choose accordingly. And then what's interesting is that, say initially you thought you had all these, but then you can still go into research, and then it's okay to leave that and shift your career, because a lot of people figure out that after some time into research, maybe this is not right, but then they do not think of what they can do beyond that. But they can do a lot of things, because there are a lot of skills you develop during this journey of training. So you will have an analytical and critical mindset. You will be able to logically think and understand. You will get a lot of management skills of managing a lot of people, managing your lab, interacting with people, managing yourself, organizing your day, organizing your work and everything. Then you do a lot of writing. you have to communicate, you have to publish your papers, you get writing skills, and then you have communication and networking skill. You present, you go to conferences, you interact with people, and then you also sometimes become audacious because you have the confidence to take risks. So that all of these are developed during the training. So this, I can go on with more of these. So many of these are very suitable for a lot of other industries and other careers. So even if after your research you are still not convinced you did the right choice, it's okay to actually shift careers. So this is for PhD research scholars who took it and then they are not comfortable. After your PhD, still do explore other options. For MTech students, do make sure you have this. You think you suit here in this bucket, only then you go for a PhD. Just don't go for the sake of a degree. Yeah, and for BTech students, you still have time to explore. So you can still try MTech and then decide, or then you can still go on for PhD and then decide. That's what I wanted to tell everyone. And before I conclude, I should surely thank my mentors, my thesis advisors, Dr. Aditya Murthy at Center of Neuroscience in Indian Institute of Science, and Dr. Radhakant Pati, Department of Aerospace Engineering, ISC, for their mentoring, training me into a good researcher. I have to surely thank Dr. Angeline Kuriba from SRM University. She was my MTech project coordinator, and it was her motivation and support that helped me do collaborative project with Dr. Srinivas Chakravarti at IIT Madras, and that introduced me into the field of neuroscience, and I'm very happy about my choice. And I also have to thank Dr. Minimol B at my undergraduate college, Model Engineering College. She actually motivated me a lot. She was the cornerstone in a lot of my career choices, especially towards research. So I thank all of them, and I also thank the audience for being patient and listening to me blabbering for an hour. I can take questions if there are.
Varsha, actually, Angel ma'am is sitting with me. We are sharing a now.
Hi, Varsha.
Hi, ma'am.
Nice to see you. Actually, we are proud of you people, alumni people settled in different concerns, companies, and you are working very good research in different concerns. Actually, if we speak physiological modeling, if we take not even a single batch of students are not doing physiological modeling. The real thing is because most of the expectation, they are clinical trials, they are expectation. So now you have given some idea, you have explained depth of information in eye movement, saccadic eye movement, and how it can be implemented, how it can be tested, what is the application and all. You have given a very depth of information.